Now that you've had a bit of a break, let's continue learning about chromosomes. As I mentioned before, during interphase, and specifically in the S phase of interphase, the chromosomes begin to replicate. In order to replicate, chromosomes must have three regions within each structure. The first of these is the replication origin. Chromosomes may contain several replication origins on the chromosome. This is where the process of replication will begin. These replication origins form replication bubbles that we will discuss in later chapters when we talk about DNA replication. The next region that all linear chromosomes must contain are known as telomeres. During replication, not all of the chromosome is replicated each time. To prevent loss of genes during this process, our chromosomes have evolved to contain sections of DNA at each end of the chromosome, known as telomeres. These stretches of DNA do not code for anything and simply act as a protective barrier against information loss. Research has shown that the shortening of these telomeres is associated with the process of aging. Telomere shortening has also been implicated in the development of cancerous cells. The last structure needed by chromosomes is the centromere. The centromere is only visible when chromosomes are condensed. Centromeres are important because they will allow the chromosomes to attach the mitotic spindle during cell division so that each daughter cell ends up with the correct number of chromosomes. Now let's talk about how these chromosomes are actually condensed. The process of condensation yields very tightly wound DNA protein complexes. For example, chromosome number 22 contains 48 million base pairs. During interphase, this chromosome measures approximately 1.5 centimeters long. However, during mitosis, the same chromosome measures only 2 micrometers or 0.0002 centimeters long. After mitosis has completed, the chromosome will extend back into the 1.5 centimeter length. While chromosomes are much more condensed during mitosis, there is a misconception that chromosomes are not condensed at all during interphase. This is not true. Chromosomes are always at some level of folding. Throughout the cell cycle, the DNA must be accessible to be read for translation and transcription, but must be kept within the nucleus in an orderly manner. This requires the cell to keep the chromosomes at some level of folding always. The chromosome condenses tightly only when the cell is going into mitosis. There are two classes of proteins that participate in condensation, histones and non-histones. Together, these proteins bind to DNA to form the structure known as chromatin. In this chapter, we are not going to discuss non-histones, but I want you to be aware that not every protein in chromatin is a histone. Histones are a major component of DNA condensation and perform most of the major folding patterns in the first few levels of folding. On this slide, you can see some images of histone subunits. Each of the subunits is very small and has an overall positive charge. This is a result of the positively charged R groups within the protein structure. This is important because DNA is negatively charged. This attraction of the DNA to the protein is of utmost importance. Each of these histones have a tail that enables covalent bonding at further levels of folding. The sequences in the DNA, or genes, that code for histone proteins are highly conserved across all eukaryotes. This means that almost every eukaryotic organism has identical histones. This is important to note for a couple of reasons. The first goes back to our chat earlier about similarities in genomes. If the protein structure is highly conserved across the huge domain of eukarya, it means that the structure is highly important, and throughout evolution, mutations in the histone composition and the resultant DNA folding was negatively selected against, meaning that the structure is so important that any changes could be fatal to an organism. The second reason to note the conservation is for research based around these proteins. Do you remember which model organism we would be likely to choose to study histones in? Hopefully you remember that brewer's yeast is our lowest eukaryotic model organism. Given that these sequences are so similar, I should be able to do preliminary research in yeast as a proof of concept assay before working in a more expensive and difficult model. When DNA is gently unfolded in a laboratory setting, we can see the first level of DNA folding called nucleosomes, which creates a beaded necklace-like structure. To be consistent, let's define a few structures. A nucleosome core particle is a structure that is limited to the DNA sequence wrapped around an octameric, meaning 8 proteins, structure at the core. Between each core, right here, is a variable in length sequence of DNA known as linker DNA. This linker DNA is not standardized and can vary greatly between each core particle. Technically, a nucleosome consists of one core particle plus one adjacent linker. However, colloquially, most scientists use the term nucleosome to describe the core particle alone. In this image, you can see several nucleosome core particles with linker strands between each core particle. The formation of one nucleosome folds or condenses the DNA into approximately 1/3 of the original length of the DNA sequence. After the first level of DNA folding, which consists of the protein DNA nucleosome core particle, comes the 30 nanometer chromatin fiber. These fibers are where the nucleosomes are packed into a fiber that is approximately 30 nanometers across. This packaging is facilitated with another histone known as histone H1. Histone H1 helps pull the nucleosomes together to form this next level of chromosome structure. Beyond the 30 nanometer fiber are several more levels of DNA folding that you can see highlighted on the slide above. Unfortunately, we still don't fully understand the mechanisms of the DNA folding beyond the 30 nanometer fiber. We understand that these folding patterns involve some loops and similar structures that we see in the beads on a string and the 30 nanometer fiber. But beyond that, we have not elucidated the true folding mechanisms. Let's talk about the exceptions to the rule. Most DNA follows the protein DNA packing patterns we have been discussing. However, there are some sequences of DNA that are always tightly condensed. This is known as heterochromatin. Heterochromatin consists of about 10% of the chromosome. It is usually located either near the centromere, which is the center of the chromosome, or near the telomeres, which is at the end of the chromosomes. These regions of the chromosomes are so tightly packed that most of the DNA sequence within these regions do not code for anything, and in fact, if a gene is accidentally packed into the chromatin region, it will never be expressed in the cell. An example of chromatin are bar bodies. In female organisms, some somatic cells consist of 2 X chromosomes. This duplication of a sex chromosome could lead to a variety of problems within the cell. To combat this, cells in early embryonic development determine which X chromosome is going to be utilized within the cell and which will be turned into tightly condensed heterochromatin structure. This X chromosome becomes what is known as a bar body. This X chromosome will then never be utilized by the cell for the remainder of that cell's life. An easy visual way to understand bar bodies is a calico or tortoiseshell cat. In this example, the gene coding for fur color in cats is located on the X chromosome. This means that female cats have the potential for 2 different fur colors. The cells will arbitrarily choose which of the X chromosomes to express and which to condense into heterochromatin. When cells randomly condense different X chromosomes, the different sections will create regions of fur that are orange and regions of fur that are black or white. This is why calico or tortoiseshell cats are not normally male. Male tortoiseshell or calico cats only arise when the cat has an extra X chromosome, giving them 2 X chromosomes and a Y chromosome instead of the normal 1 X and 1 Y chromosome. Let's now discuss how chromosome structures are regulated. Structures are regulated in 2 ways: chromatin remodeling complexes and chemical modification of chromatin. Let's first look at chromatin remodeling complexes. Chromatin remodeling complexes utilize ATP to loosen specific segments of folded DNA. This loosening exposes the DNA to DNA-binding proteins such as those involved in transcription. This allows the needed DNA sequence segments to be read and either transcribed or replicated. The important aspects of the chromatin remodeling complexes are that these complexes are protein machines that utilize ATP hydrolysis to perform this work. If you need a further refresher on protein machines, check out the end of Chapter 4 in your textbook. The second form of chromosome structure regulation is through chemical modification to the chromatin. Remember, chromatin is the protein DNA complex. There are 3 main types of chemical modification. All of these modifications are similar in process. These are covalent modifications that are made to the tails of the histones. The first chemical modification is one of the most well-known. This is the addition of a methyl group to the chromatin structure. This modification has been studied intensively in the cancer research field. Researchers have found that by removing methyl groups using a targeted chemotherapy, they can actually cure the cancer with little side effects. I've put a Nova video about this on the Blackboard site. The next chemical modification is an acetyl group. The last chemical modification is the addition of a phosphate group. The pattern in the modifications and the amount of modification to the histone tail can dictate how the segment of chromatin is going to be treated by the cell. Unfortunately, we still do not understand the implications of the tags and the patterns of tags. This growing field has been named epigenetics, meaning above the genome, since these modifications occur on a level above the actual coding. The primary focus of this field has been on the methyl modifications to the DNA due to interest in cancer. The take-home message on these chemical modifications is that it is a similar process to the covalent modifications to proteins that we discussed in Chapter 4. Researchers are still working to understand how these modifications occur, how permanent they are, what downstream effect they create, and how we can pharmacologically intervene on harmful modifications. Besides the Nova video, I have posted an epigenetics video in the supplemental video section that discusses the basics of this field, if you'd like to see a little bit more about epigenetics. All of this circles back around to inheritance patterns. This regulation can be passed on from parent to child in ways that we have not completely understood yet. This inheritance can be from one cell generation to the next, or between generations of organisms. Some researchers believe that a mixture of our parents' epigenetic modifications will be passed on to offspring, while others are researching the possibility that some epigenetic inheritance can take over the remainder of the DNA and override the programming of the other parent. Either way, the ability to inherit the specific chromatin structure helps ensure that eukaryotic cells remember what they are supposed to do, or what cell type they are going to be. This ensures that we look and have similar functions to those that we are descended from, and sets our progeny up for survival in future generations. This is the end of Chapter 5. I hope most of this material is review for you, and that we are only adding to the picture that you already had about DNA and chromosomes. In fact, for most of our chapters, this is what I expect. Essentially, all we're doing is filling in some of the finer details to the broad foundation of knowledge that you received in previous courses. As always, if you have questions, please let me know and we will discuss them either by office hours or over emails, or in class.